We'll take a closer look at the brain. So the brain itself is a relatively large structure, and so it weighs approximately 3 pounds in the adult. So it takes up quite a bit of space and weighs quite a bit. The majority of the brain itself, as we know, is made up of the glial cells, and then we have the neurons and all of their connections that are there as well. And depending upon the connections that are made, is going to determine the ability to remember, contemplate, and work within the confines of the brain itself. And so the actual ability to make new connections and create those connections is what derives the intelligence of a person as well. Within the nervous system, we have the ability to take all of the stimuli that is coming in and determine what we need to do with it and move on from there as far as an output that is there. Many neurons in terms of the brain itself, creating many even more components as far as the number of connections that are between each cell itself. And so just a huge amount of computing power within the neurons that we have there. And so here we can see a view from the inferior aspect of the brain. We can see all of the areas where we're completing the processes of intellect in terms of the cerebrum itself. We have the brainstem continuing on down from the diencephalon. And then we have the cerebellum kind of hanging out behind the brainstem. And then on this one as well, we can then see all of the cranial nerves that are associated with the brainstem as well. And so the first two cranial nerves that we have, the first one being the olfactory here, arising from the olfactory bulb and continuing down into the nasal cavity. We also have the optic nerve, and so we have the nerve that brings us our sight there. Those are the only two that aren't actually associated with the brainstem itself. All of the other cranial nerves are actually originating from the brainstem. And so only the first two of the 12 do not actually originate from the brainstem itself. All the others do make their origin and have their nuclei within the brainstem. So the brain tissue itself, when we look at the brain, what we essentially see when we first open up the brain is going to be gray matter. And so gray matter is on the outside of the cerebrum and white matter is on the inside. And so this is where we have all of our cell bodies. We have unmyelinated fibers. We have our glial cells that are all within the gray matter itself. And this is making kind of the outermost cortical portion, the cortex of the cerebrum itself, is all of the gray matter, including that of even the cerebellum and the brainstem. We have all of our gray matter on the outside. The white matter is on the inside, and so we're going to see it tucked in between the gray matter that is there. This is primarily all myelinated fibers. And so this is connections, axons going from one place to the next within the cerebrum here. We do have one special area of white matter as well called the corpus callosum. This is where that white matter is going to be, in a sense, tracking from one side to the other. And so we have a way to cross the two hemispheres themselves and get information to cross from one side to the other when needed there as well. Even within the white matter, though, we do have some areas of gray matter nuclei. So there are some areas that are going to be gray matter inside there as well. And so it's not all just white matter that we find within the white matter areas themselves. The brain, as it sits inside the skull, is pretty protected where it is. And in order to keep it that way, we have a whole bunch of structures that are going to help with that. Everything from the hair on the top of your head to the skin, moving into the structures within the skull itself. And so we get the bones that are there. for the skull, creating a strong, hard structure for them to be held within. And then just deep to the bones, we find the meninges. And so we have this protective connective tissue that is going to surround the brain itself. And we'll talk about those in just a little bit. In between the layers of the meninges, we then have cerebrospinal fluid. And so we have a fluid that is going to essentially help to float the brain and allow for the brain to essentially decrease its mass, or not its mass, but its weight within the skull. And so it kind of lightens it and allows it to be cushioned inside there, protecting it from when we're moving our head side to side, up and down, banging it against the wall, etc. All of that helps to protect the brain that is there. And then the last kind of component, which is helped by the glial cells themselves, is the blood-brain barrier. And so this is going to keep any kind of harmful substances out of the blood, or that may be in the blood, from getting into the brain matter themselves. So those meninges come in one of three kinds. We either have dura mater, arachnoid matter, or pia mater. And that dura mater is the outermost covering. And then we have the arachnoid matter, and then we have the pia mater. This has a large number of blood vessels that run in and along the meninges themselves. This allows us to have that fluid layer there surrounding the brain as well. And so we have our CSF, our cerebral spinal fluid, that is there as well. The meninges also, in terms of that CSF, help with the draining of the CSF away from the brain and bringing it back towards the venous system, and allows us to get it out of the brain itself. So the first layer, our dura mater, is really tough. It's almost leather-like. And so it's a very tough layer. It has two parts to it. It has a periosteal layer, and then it has a meningeal layer. The periosteal layer is the portion that is right next to the bone. And so that's going to be right next to the periosteum of the bone. And then the deeper layer, the meningeal layer, is the portion that is, in a sense, next to the brain. Deep to the dura, deep to the dura mater, we have the subdural space. And so we have a little bit of a space that lies in between the dura mater and our next layer, the middle layer, our arachnoid mater. So here we have that subdural space that is in between the arachnoid and the dura. And so there's a little bit of a space there in between the two that is there. We're going to have some blood vessels in those areas there as well. Deep to the arachnoid, which is our next layer in, and on this particular picture is this portion here, but it also includes all of what almost looks like a little net here. And this is what is creating part of the arachnoid and part of its ability to kind of hold on down to the pia mater below. The arachnoid also has little extensions called arachnoid villi, and these allow for the fluid from the CSF to move out of the CSF and into the veins. And so this starts to get us into the venous system, and in this particular portion here, into the dural venous sinus. That's there. And so all of the web-like portions here are the trabeculae. And so similar to that of like the bones, we have trabeculae, kind of this network that is inside there, creating that space in between the arachnoid and the pia. And so we then have a space there, the subarachnoid space, that lies between the arachnoid and the pia mater. The pia mater is the deepest layer, and so the pia mater is intimately touching the brain itself, and so this is touching the cortex itself there. When we follow the dura mater and the arachnoid mater around the brain, we'll find that the dura mater and arachnoid mater just kind of cover over the surface in a sense. They don't go down into the sulci. They don't go down into the fissures of the brain as much, or they will go down into the fissures, but not into the sulci. And so they'll make their way down into the larger fissures, but not into the sulci. And that versus the pia mater does actually go down into all of the individual sulci. So it's getting down into all of the tiny little spaces within the brain itself that's there. The dura mater creates kind of these little infoldings that comes together when two sides start to meet one another. And so the biggest one is the falx cerebri. And so this is this big, almost looks like a sail here that goes from the anterior to the posterior part. This is running in between the two hemispheres themselves. So this is down in the longitudinal fissure of the brain. This is going to separate the two. It contains the superior sagittal sinus. So we've got a big blood vessel that runs along the length that is there. And then we have the inferior sagittal sinus as well, running along the falx cerebri. The cerebellum has a falx as well. This is the falx cerebelli. This essentially separates the two halves of the cerebellum as well. And then lastly, and this picture doesn't really show it too well, we have a little piece that kind of sticks out right in here, which becomes the tentorium cerebelli. And this is going to separate the cerebrum from the cerebellum. Deep within the brain, we have the ventricles. So as the brain was developing from the neural tube developed into the primary vesicles and went from there. And that space that was within the neural tube, in a sense, kind of stays there. And so that develops into the brain ventricles. The brain ventricles, we have the lateral ventricles that are there. The lateral ventricles themselves have kind of three major portions to them. And so they have an anterior horn right in there. They have a posterior horn, and then they have an inferior horn. So these are kind of C-shaped structures with an anterior horn, an inferior horn, and then the posterior horn would be coming out this way. And so we have three different portions to it. Those lateral ventricles drain down into the third ventricle, that is here. And then as they get into the third ventricle itself is separated by the two halves of the diencephalon. And so if we kind of look at where the thalamus is on either side, in a sense, the space in between where the thalamus would be is that third ventricle. So all of this space inside here is the third ventricle itself. We can see actually where that brain portion here comes in, and we have an empty spot in that third ventricle. This is where the thalamic adhesion is located within the diencephalon itself. The third ventricle then drains down through the cerebral aqueduct, and then that gets us into the fourth ventricle, which then lies primarily between the pons and the cerebellum, and then a little bit of the cerebellum and the medulla oblongata. And then from there, it drains down through the central canal of the spinal cord. So cerebrospinal fluid itself is what's going to be contained within those ventricles. And inside those ventricles, we have all of the nutrients and all of the oxygen and everything that the brain is going to need. And then it eventually gives up its waste product to it as well. And that fluid is essentially looking clear like water. And so it's kind of like the plasma that we have in the blood, except that this fluid has been filtered in a sense. And so it's been filtered by the choroid plexus. And the choroid plexus, along with the ependymal cells, are going to generate this fluid. And so this is going to create the fluid from the blood plasma and only allow, in a sense, the good things into the brain's exposure that is there. As the brain floats inside that cerebrospinal fluid, it gives it buoyancy. And so it's going to kind of float it inside the brain. When somebody gets a spinal tap to draw some of that fluid out, their brain will typically sink inside the cranium, and they will oftentimes get a pretty good headache with it. So it does provide for protection. So as it floats inside there, it's not going to bang around quite so much. And then with all of that fluid, we have a nice stable environment as far as the chemicals go that are surrounding the brain itself. So in order to maintain our blood-brain barrier and only allow certain substances to cross that blood-brain barrier, this is where we need to make sure that different substances that may be found within the bloodstream don't get to the brain tissues themselves. And so certain drugs or chemicals won't be able to cross that blood-brain barrier. Other waste products in the body won't be able to cross that blood-brain barrier that's there. Certain substances that we need to monitor, things like ions and hormones and things like that, will cross that blood-brain barrier, and the blood-brain barrier does break down in a couple of different places. And so we don't have it kind of everywhere, but we do have where we have to be able to make the CSF. We have the choroid plexus. We have it around the hypothalamus as it monitors things that are going on within the body itself, and then at the pineal gland as well. And so this is going to take that ability to limit the amount of substances that are there. And this is where, once again, those astrocytes come into play in that they surround the blood vessels. They're going to maintain that barrier there to ensure that only certain substances are able to come through the capillaries themselves. So they're going to create this kind of extra covering to the capillaries themselves and limit what can get through. When we look at the brain itself, we have some relatively distinct regions that we can see. And so we have the cerebrum. We then have the diencephalon. And then we have the cerebellum. Extending downward from the diencephalon, we then have the brainstem as well. And so those are our distinct areas that are there. The brainstem is associated with all of those 12 cranial nerves that we mentioned at the very beginning. That's there. The brain itself is a relatively large structure, as was mentioned, and with that, we need to be able to increase the surface area of the brain, and in order to do that, we're going to have infolding. So the brain is going to fold in on itself, creating gyri and sulci. So the gyri being the bumps, the sulci being the grooves, and this allows us to increase the amount of cells that we have available. And so we're going to increase that ability to have more and more neurons available for the normal processing. And so when we look at the hemisphere, the two hemispheres themselves are divided by the longitudinal fissure coming down the center of the brain, and that's going to divide us into a right and a left hemisphere that's there. From there, we have the frontal lobe, and then the parietal lobe. Behind the parietal lobe to the posterior, we then have the occipital lobe. That is there. The frontal lobe and the parietal lobe are separated by a central sulcus. This central sulcus is a unique sulcus in that it's the only one that goes uninterrupted from the longitudinal fissure all the way down to the lateral sulcus. And so it's going to be uninterrupted on there. We then have another sulcus that's oftentimes a little bit difficult to see, the parieto-occipital sulcus in between the occipital lobe and the parietal lobe. Within this cerebrum, this is in a sense everything that makes us us. And so everything that is from intellect to your personality, to your ethics, to how you do things, to your motor skills, to your sensory ability to detect things, is all out here in the cerebrum itself. So the central sulcus, as I said, separates the frontal lobe from the parietal lobe. And so it runs down here from the longitudinal fissure all the way to the lateral sulcus uninterrupted as it makes its way down there. It also separates between motor function on the anterior side and sensory function on the posterior side. So the precentral gyrus here is motor, and the postcentral gyrus is sensory. And so we're going to separate that information that is there. And so that motor information that is going out, this is what's going to end up going down towards the cord. The sensory information that is coming into the cerebrum here is the initial stimulus itself. It tells us where on the body it occurred. It tells us exactly what happened in terms of the type of information, but then it also has to go to the somatosensory association area to determine what we think of the information that came in, to put it all together with our past experiences and allow us to form essentially a bit of a conclusion as to what type of information was there for that. A large portion of the frontal lobe is all in doing things like our intellect, doing things like our emotions, doing things like our personality, and many of them are found within the limbic system itself. When we look at the cerebrum and specifically look at the precentral gyrus and the postcentral gyrus, this is one of the kind of interesting things that comes with the brain, is that we have what we call somatotopy. And so this somatotopy that we have is that we have essentially a caricature drawing of the body on the cerebrum. And so when we look at it, we can see that both the sensory and the motor side, we don't really have equal representation of the body parts. And so our face, our head, is disproportionately large compared to, say, our torso. And so those areas there are very askew. Even our hand is almost as big as our face. Our foot is relatively large. And what it comes down to is those areas that have more neural input, both on the motor side and the sensory side, are going to have more areas of the cortex dedicated to them. And so those areas that we have the ability to do more fine motor control, we have more cortex dedicated to them. Those areas where we have more sensory input, we have more cortex dedicated to them. And so we can both also take together the fact that whenever we have a large number of motor neurons innervating an area of the body, we're also going to have a large number of sensory neurons innervating that portion of the body as well. The diencephalon is kind of a deep portion of the brain. And so this lies deep, deep, deep inside the brain. This contains three different major regions. We have the thalamus, the hypothalamus, and the epithalamus inside there. And so the thalamus is kind of this egg-looking portion here. It has the interthalamic adhesion there as well. The hypothalamus is this little front portion here. And then the epithalamus is just a little portion here and back for those. The hypothalamus and the thalamus are kind of big players within the entire body itself. And so the hypothalamus has essentially a hand in all of our internal homeostatic balances. And so this is essentially where homeostasis is maintained, is within the hypothalamus. And so that's our master control for basically everything homeostasis-wise within the body itself. From there, we have the thalamus. And the thalamus is kind of the most important part when it comes to sensory information coming up to the cortex. All sensory information is going to make a stop in the thalamus and then head up to the cortex from there. Motor information also travels down through the thalamus here as well. And so we're going to have motor input traveling through here also. The epithalamus is a small little region here towards the back. It does have a structure that sticks off of it called the pineal gland or the pineal body. And this little guy here is responsible for the production of melatonin. And so melatonin is a hormone that is important for our sleep-wake cycles. And so this is what helps us to determine when it's time to go to sleep, when it's time to wake up. And so what do we need to do in terms of that process that's there? When should we wake up? When should we go to sleep? Is determined by the relationship of melatonin levels throughout the day. And so those levels go up and down throughout the day. And that's what makes us sleepy or makes us want to wake up on there. The mammillary bodies, which are in a sense kind of sticking off of the between the hypothalamus and the brainstem, are what's going to help with kind of feeding reflexes, especially when it comes to smell. So our brainstem itself here, we have an anterior view of the brain, and we can see that the optic nerves, as they pass, it would be coming out through here, as they pass around the brainstem there, are just kind of going along for the ride. They're not actually making any contact with the brainstem itself. And then we have our oculomotor nerves, the first of the three nerves that are going to extend off of the brainstem itself there. This picture here is a little bit easier one to see and appreciate those mammillary bodies as well. And so two little bumps there. This happens to correlate with our sense of smell. So other animals that rely more on their sense of smell are going to have much larger mammillary bodies that are there. The brainstem has all the nuclei for each of our cranial nerves, and this is going to process everything from reflexes for vision and hearing and balance and smell and digestion. All gets processed within the brainstem itself. That's there. And the majority of those visual and auditory reflexes, kind of putting the two together, occurs within the corpora quadrigemini. A little bit next to that, in a sense, and a little bit lower, we have the red nucleus. And the red nucleus and the substantia nigra are both going to extend out from the pons there. And as they do so, we have the ability to control many of our motor functions. And so within the red nucleus, this is going to control motor commands that are essentially unconscious for postural control. And so kind of like keeping your head above your shoulders above your hips, and your hips above your feet. In that process there, if you're just standing, you don't think about the tiny little motor control changes that have to occur in order to keep you from falling over once it's there. The substantia nigra then is working within motor input to work with keeping muscles, in a sense, quiet when we don't want to use them. And so one of the big things there is that process. And so here we have those particular areas. Here we can see the red nucleus. We can see the substantia nigra. That is there as well. One of the other, in a sense, kind of oddities, so to speak, as far as the cranial nerves goes, is that we have the trochlear nerve here, originates from the posterior side of the brainstem. And so it emerges from the brainstem along that posterior side. Down in the pons here, we can see that the pons is pushed forward by the cerebellum. And so it kind of makes it stick out there a little bit because the cerebellum is present there as well. So the majority of all the pons is kind of bulging anteriorly once it's there. The pons works in terms of respiration, and that it's going to be one of our inhibiting centers for respiration. So it's going to control when we basically shut off our respiration. So it's going to control the rate and the depth of inspiration itself. The cerebellum is important in the aspects that one of the big things that it does is that it controls our unconscious motor control. And so it has sensory information that comes into it. It has this big, large amount of white matter that spreads out like a tree, hence its name arbor vitae. So it's going all over inside there. And it spreads all throughout there. Large amount of connections occurring within all of that white matter that is there, and a large amount of cell bodies. So a lot of gray matter there as well. It's receiving unconscious proprioceptive information about all of our joints, all of our muscles, all of our skin, and it's essentially kind of fine-tuning motor control. And so it's anticipating what we want to do and whether we're going to be able to do it or not with our current kind of processing that we have at that moment. And so for where we are we able to maintain that control? The cerebellum also controls cognitive information, does a lot of problem solving for us as well. And the cerebellum is one of those areas that is susceptible to a chemical that can cross the blood-brain barrier, and this would be alcohol. And so this is part of the reason why, since it's always involved in equilibrium, this is why when somebody is suspected of having drank too much, they're going to make them do balance types of tests in order to determine whether the cerebellum has been affected too much or not. The last portion of the brain in terms of the brain stem is the medulla oblongata. And so the medulla oblongata is the portion that is going to connect us down to the spinal cord. And so it's going to make that connection on its way down. This one is kind of the rest of our internal organ functions. And so this is both cardiovascular and respiratory as far as the controls that it has working for it. And so this is rate and rhythm of the cardiac muscle. This is blood pressure control via smooth muscle in our blood vessels. This is reflexes for respiration, things like coughing and sneezing, but also reflexes for digestion, and so salivation and swallowing, as well. If something gets stuck into the back of the throat, we may end up gagging and or vomiting as well. And then the last of our cranial nerves are associated with the brain stem here in the medulla oblongata as well.